Hello friends, myself Dr. Ravi Manek, working as assistant professor from BK Modi Government Pharmacy College, and today purpose of making this video lecture is to prepare the students for the competitive exams like DI exam, which is going to be held or scheduled in few months. So topic of this video lecture is pharmacokinetics. As we know that pharmacokinetics is the process which gives you idea about what body does to the drug. Body absorbs the drug, body distributes the drug, body metabolizes the drug, and body excretes the drug. And during all this process, means ADME process, means absorption, distribution, metabolism and excretion, drug has to cross the cell membrane. So one of the important aspect is drug transport. Now we will have idea about drug transport, which are the various mechanism for the drug transport, and we will have idea about the sample questions. So here I have taken one sample question of senior drug inspector exam 2020-21. One thing I would like to highlight prior to getting entry into the topic, that is here we have taken 5 previous question papers of GPSC exams of DI, senior DI and other examination as a reference, and here we will focus on solving these previous questions. So this question I have taken from SDI exam 2021. Question is, which of the following transport mechanism is independent of carrier protein molecule? Options are primary active transport, secondary active transport, passive diffusion and facilitated diffusion. Now if you recall that the answer of this question is passive diffusion. Now if you see the justification, for justification we would have a brief idea about the drug transport. Generally, small drug molecules crosses the cell membrane by 4 different ways. It may cross by 4 different ways. First, by diffusing through lipid, that is a passive diffusion. Second, by diffusing through aquaporins. These aquaporins are special transport protein that will traverse the lipid. Third, carrier mediated transport system. Here the drug combined with the transmembrane carrier protein, it binds the molecule on the one side of membrane and then changes the conformation, and it releases on other side. And fourth one is pinocytosis. It is also known as cell drinking. So this is diagrammatical representation. Drug diffusion through the lipid, drug diffusion through aquaporins, carrier mediated transport system. Now carrier mediated transport system, it is a specialized transport system which is meant for the physiologically important molecules like sugars, amino acid, neurotransmitters and metal ions. Here these physiologically important molecules bind with the carrier molecule, and it will undergo the conformational change, and these molecules are released on other side of the membrane. Now if it is performed purely passively, means if no energy is required or without usage of any energy source, then it is known as facilitated diffusion. These facilitate the process of transmembrane equilibration of the transported species or the drug molecule in the direction of electrochemical gradient. So it will work in the direction of electrochemical gradient of the transported species, that is facilitated diffusion. Second one is active transport. Here electrochemical gradient of sodium is utilized. In this case, transport can occur against the electrochemical gradient of drug molecule or transported species. Now, further active transport may be classified into primary active transport and secondary active transport. Now, the characteristic of carrier mediated transport system is that it may get saturated because there is a binding step. This binding site may be occupied and it will undergo the saturation. Second thing, there is characteristic of competitive inhibition. If there are two drug molecule which binds with the same carrier protein, so they will compete with each other, and one drug molecule will inhibit the transport of another drug molecule, and it is ubiquitous, and many pharmacological effects are the result of interference with them. So from pharmacokinetic point of view, though there are very few sites in the body where carrier mediated drug transport system is important, but important ones are renal tubule, biliary tract, blood brain barrier, and GI tract. If we look into secondary active transport, there is a slight difference as compared to the primary active transport. In secondary active transport, ATP is not directly coupled to the molecule of interest. Instead, another molecule is moved up its concentration gradient, like sodium. It will generate the electrochemical gradient. The molecule of interest is then transported down to the electrochemical gradient. As this process still consumes the ATP to generate the gradient, the energy is not directly used to move the molecule across the membrane, so it is known as secondary active transport. It utilizes the co-transporters. Co-transporters may be of two types, antiporters and symporters. Depending on the whether the substance move in the same direction or opposite direction. A name itself suggests if substance is moved in opposite direction, then it is antiporters, and if it is a substance is moved in the same direction, then symporters. Here is the diagrammatic representation. Here you can see the uniport. A symport means both the substance are transported into the same direction, and antiport means both the substance are transported in opposite direction. These are the examples of transporters. So now we will try to solve other questions. This question is from JC exam 2021. Which transport system covers the symporters and antiporters? Options are A, primary active transport, B, secondary active transport, C, passive diffusion, and D, endocytosis. So now you may be easily able to answer this question. It is secondary active transport, because in case of secondary active transport, the drug molecule will utilize a molecule which is transported through ATPase pump, and then the drug molecule is transported against the electrochemical gradient. Another question is, movement of water across the membrane is facilitated by proteins called option A, annexin, option B, aquaporins, option C, selectins, and option C, transportins. So we have seen in very first slide of the discussion and second slide in diagram also, we have seen that there are aquaporins which transports the drug molecule along with the water. So answer of this question is aquaporins. Next question, which of the following process is also known as cell drinking? In very first slide we have seen that pinocytosis is the process in which it is also known as cell drinking. the type of membrane transport that uses the ion gradient as energy source. Options are facilitated diffusion, B passive diffusion, C primary active transport, D secondary active transport. Answer is secondary active transport. Here ion gradient is used as the energy source. In case of facilitated diffusion, there is no energy is utilized. In case of passive diffusion, there is no energy is utilized. In case of primary active transport, the drug is transported along with its own electrochemical gradient or against its own electrochemical gradient, but it will utilize the ATP. And in case of secondary active transport, it will utilize the ion gradient. It will utilize the ATP, which is associated with the ion gradient, and the drug will transport it either in same direction or in opposite direction, means antiport and symport. One more fundamental is, see, all this previous process which we have seen about the drug transport is for the small molecules. If the drug molecule is too large to directly pass through the lipid bilayer of the membrane, then it will undergo the endocytosis or exocytosis process. So large molecules, microorganisms and waste product are some of the substance which are moved through the cell membrane via exocytosis and endocytosis. If it is taken into the cell, then this process is known as endocytosis, and if it is thrown outside the cell, then it is known as exocytosis. Endocytosis may be of two types, pinocytosis and phagocytosis. Smaller molecules are transported through the pinocytosis, and larger molecules are transported through the phagocytosis. Exocytosis involves the passage of vesicle from the endoplasmic reticulum or Golgi apparatus through the cytoplasm to the cell membrane, where it fuses and releases its content. So here difference between endocytosis and exocytosis has been shown. Endocytosis is the process of taking the particle or substance from outside the cell and transferring it to the inside using the vesicles, while exocytosis is process of sending the substance or a particle from the cell, inside of the cell, to the outside the cell using vesicles. Major functions which are done by endocytosis are absorbing the nutrients for cell function, eliminating the pathogens, disposing of the old damaged cell. And major of the function which is performed by exocytosis process is removing toxins or waste product, repairing the cell membrane, facilitating the communication between the cell. Types of endocytosis is it is of two types, phagocytosis and pinocytosis, while exocytosis is of two types, regulated exocytosis and constitutive exocytosis. Examples: white blood cell engulfing a virus and eliminating it by endocytosis, or releasing the neurotransmitter of for the cell communication is done by exocytosis. So now let's see the sample questions. Which kind of drugs are absorbed through the endocytosis? So now you know very well that larger drug molecules like macromolecular drugs are absorbed through the endocytosis. So in all these four options, that is option A, molecular weight ranging from 100 to 400 Dalton, option B, water soluble drugs, option C, macromolecular drugs or drugs oily droplet, and option D, polar drugs. Out of these four options, you might be aware now that option C is right. Macromolecular drugs or oily droplets, drugs as oily droplets are absorbed through the endocytosis. Which of the following process is called cell drinking? It is pinocytosis. Option A, pinocytosis, is the correct answer. It is a type of endocytosis process where small molecules are transported through this process. Second one is absorption of poliovirus from GI tract takes place via option A pinocytosis, option B passive diffusion, option C phagocytosis, and none of the above. So correct answer is pinocytosis. Now let's see the absorption process. Simple question is mostly weakly basic drugs are absorbed from A intestine, B stomach, C stomach and intestine, and D cannot be absorbed. And correct answer is stomach. Now let's see the fundamentals. Most of the drugs which are taken by mouth and it is swallowed, they are absorbed through the intestine, and mechanism of the drug absorption in the intestine is passive transfer. It is absorbed through the passive transfer. Most of the drugs are absorbed through the passive transfer, and these rate of this reaction or rate of this passive transfer is determined by the degree of ionization and lipid solubility of the drug molecule. As expected, strong bases of pKa 10 or higher are poorly absorbed because degree of ionization, and strong acids of pKa less than 3, they are also poorly absorbed because they are fully ionized. In both the cases, degree of ionization is very high, so they are fully ionized, and we know that ionized species are impermeable, or they are not able to cross the cell membrane. So here example of curare poison has been discussed. Curare poison was used previously by South American Indian as a arrow poison. It is containing quaternary ammonium compounds which blocks the neuromuscular transmission. So it is a strong base. It is poorly absorbed from the GI tract, so the meat from the animals which is killed by this curare poison is safe to eat. It will not get absorbed because of the ionization. Now there are certain exceptions, or there are certain cases where intestinal absorption occurs through the carrier mediated transport system. Here 3-4 example has been given. A drug levodopa, which is used for the treatment of Parkinson, it is taken up by the carrier that is normally transports the phenylalanine because of the structural similarity. One more example is of 5-fluorouracil. It is a cytotoxic drug. It is transported by the system which carries the pyrimidines like thymine and uracil due to the structural similarity. Iron is absorbed by a specific carrier which is present in the jejunal mucosa, and calcium is absorbed by the means of vitamin D dependent carrier system. That's why certain protein powders or certain nutraceutical powders are highlighting that instead of drinking plain milk, you should drink the milk containing these nutraceutical product which is having vitamin D. It will help to absorb the calcium present in the milk, because calcium is absorbed by means of vitamin D dependent carrier system. Now if you see the factors affecting GI absorption, typically about 75% of the drug which is given orally, it is absorbed within 1 to 3 hours. And there are many factors which are affecting, like GI motility, splanchnic blood flow, particle size and formulation, and physicochemical factors. Now GI motility has very large effect. Certain disorders like diabetic neuropathy and migraine decreases the GI motility, so slows the drug absorption. Even drug treatment can also affect the motility by either reducing the motility or increasing the motility. The example of the drug which reduces the GI motility is muscarinic receptor blockers or anticholinergic drugs like dicyclomine. Or the example of the drug which increases the GI motility is metoclopramide. That's why it is used in the migraine to increase the absorption of analgesic. We know that in migraine GI motility is decreased. If analgesic is given for the treatment of migraine, the absorption of analgesic will be less. So if the analgesic is given along with the metoclopramide, so metoclopramide will normalize the GI motility which has been reduced by the migraine, and it will increase or facilitate the absorption of analgesic. So there will be rapid relief. Even excessive rapid movement of gut content will impair the absorption, like in the case of the diarrhea. Second factor is splanchnic blood flow. Generally, if drug is taken after meal, it will slowly absorb because it will slowly progress towards the small intestine. But after meal, splanchnic blood flow is increased, so there will be higher plasma concentration. Example is propranolol. Third factor is particle size and the formulation. It has a major effect on the absorption. For example, digoxin is poorly absorbed, but making a small difference into the pharmaceutical formulation, it will increase the extent of absorption. Certain physicochemical factors also affect the absorption, like tetracycline binds strongly to the calcium, and calcium rich food like milk will prevent its absorption. Bile acid binding resins such as cholestyramine binds with the several drug, and it will prevent the absorption, like warfarin and thyroxine. Generally, when drug is administered orally, the purpose is to produce the systemic effect, but there are exceptions like vancomycin and mesalazine. Vancomycin is poorly absorbed drug, and it is when it is administered orally to eradicate the toxin forming Clostridium difficile from the gut lumen in the patient with pseudomembranous colitis. One more example is mesalazine. It is a formulation of 5-amino salicylic acid which is in pH dependent acrylic coat, which degrades in the terminal ileum or proximal colon. Here it will produce its anti-inflammatory effect to treat the inflammatory bowel disease. Bioavailability is the term used to indicate the proportion of the drug that passes into the systemic circulation after oral administration. So it will take into the account both absorption and local metabolic degradation. So let's see the sample questions from this portion. Absorption of the tetracycline is reduced in presence of milk due to option A hydrogen bonding, option B protein binding, option C ionization, and option D chelation. We know that tetracycline has affinity for the calcium. It will get chelated, and thus milk will decrease the absorption of tetracycline. So answer is option D chelation. One more question from SDI exam 2021. Parietal cells of the stomach produce intrinsic factor which is essential for absorption of vitamin. Options are vitamin A, folic acid, vitamin C, and vitamin B12. Answer is vitamin B12. Now let's see the justification. Vitamin B12. In case of vitamin B12, body absorbs the vitamin B12 into two steps. First step, hydrochloric acid in the stomach will separate the vitamin B12 from the protein to which it is attached. And in second step, this free form of vitamin B12 is combined with the protein which is made by the stomach, and the name of this protein is intrinsic factor, and thus body will absorb them together. Now let's see the absorption of folic acid. It is absorbed by an active transport process which primarily occur into the duodenum and jejunum. Ascorbic acid, that is actively absorbed by the sodium vitamin C co-transporter. This is example of secondary active transport. It is driven by the sodium electrochemical gradient created by sodium potassium ATPase. Ascorbic acid is then diffused into the capillary and ultimately enters the general circulation. So vitamin C is generally circulates as ascorbic acid. And vitamin A, esters are removed by estrase, so the free retinol can be taken up into the enterocyte. Once provitamin A carotenoids are taken up into the enterocyte, there are two options. One, it may be cleaved to the retinal and then converted to retinol, or it is absorbed intact and incorporated into the chylomicrons. Next question, proton pump inhibitors markedly reduce the bioavailability of ketoconazole because option A, PPI decreases the gastric pH, option B, PPI increases the gastric pH, option C, PPI enhance the elimination of ketoconazole from gut, and option D, none of the above. Now we know that proton pump inhibitors are used to prevent the acid secretion. They binds with the proton pump, which is a terminal step of acid secretion, and they will decrease the acid secretion. So if we see the effect of PPI, they will increase the gastric pH. Now ketoconazole is the drug whose absorption is better in acidic aqueous solution. So the drug which alters the pH of stomach will affect the absorption of ketoconazole. Here the answer is option B, PPI increases the gastric pH, so it will reduce the bioavailability of ketoconazole because it will reduce the absorption of ketoconazole by increasing the gastric pH. The GI tract absorption of the riboflavin is more if it is taken option A, fasting state, option B, after meal, option C, after exercise, option D, none of the above. Now we know that after meal the splenic blood flow is increased, so the answer of this question is after meal. So the increased splenic blood flow will increase the bioavailability of the drug by increasing the absorption. So drug like riboflavin, they are taken after the meal. Which form of ampicillin is absorbed quickly? Anhydrous, dihydrate, trihydrate, and none of the above. Answer is anhydrous because anhydrous form has been reported to have better bioavailability than the trihydrate form, maybe due to its greater aqueous solubility. Here you can see the research has been, this research article discussed the bioavailability of ampicillin anhydrous and ampicillin trihydrate. Which of the following is absorbed most rapidly? Secobarbital, barbital, thiopental, and all of the above. Answer is thiopental because of the thiol group in the structure of thiopental, its lipid solubility has been increased, so it will get absorbed rapidly as compared to the secobarbital and barbital. Thank you for your patience to go through this video, and I hope this video will be helpful to clear the concept of drug transport and absorption, and we have seen the sample questions which has been asked previously. Thank you and best of luck.